The limiting dilution cell transplantation assay is the gold standard for assessing self-renewal potential in experimental animal models. In this demonstration, one-cell stage Syngeneic zebrafish embryos are injected with Rag2-MYC and Rag2-GFP to create transgenic zebrafish that will develop fluorescently-labeled T-cell acute lymphoblastic leukemia by 60 days of life. The primary leukemia cells are isolated from zebrafish and then purified using fluorescence-activated cell sorting. Next, the cells are transplanted at limiting dilution into the peritoneal cavity of adult zebrafish, and the fish are monitored for tumor growth for up to 90 days. Finally, the extreme limiting dilution analysis statistical software program is used to quantify the frequency of leukemia-propagating cells within the original tumor.
Limiting dilution cell transplantation analysis allows investigators to accurately assess the number of self-renewing cells contained within the bulk of the tumor mass. It is these self-renewing cells that drive cancer formation and are ultimately responsible for relapse.
The main advantage of doing limiting dilution transplantation assays in the zebrafish model is that many fish can be transplanted for each experiment, resulting in better precision when determining the frequency of self-renewing tumor-propagating cells.
This method provides insight into tumor-propagating cells in T-cell acute lymphoblastic leukemia. It can also be applied to understand other zebrafish cancer models.
Jessica Blackburn, a fellow in the lab, and Sali, a talented technician, will be demonstrating the procedure for you today.
To linearize Rag2-C-MYC and Rag2-GFP DNA constructs, digest 10 micrograms of each plasmid with restriction enzyme NOT1 at 37 degrees Celsius overnight. After phenylchloroform extraction and ethanol precipitation, resuspend each plasmid.